Concentration gradients. Okay, so this is a concept that we have to get to know. We have to understand what a gradient is. You may have seen, signs on the side of the road for trucks that say steep grade, use low gear. Grade is a gradient. And for them, the gradient is a slope from a high elevation to a low elevation, right? And here, this is a gradient, but this isn't a gradient about elevation. This is a gradient about concentration. Why do I have these two brackets here? Because this is the abbreviation for concentration. So if you're taking notes, you don't have to write concentration. If you want to talk about the concentration of oxygen, you can just say bracket oxygen. And I know that you mean concentration. Okay, so this is a difference, and we can see here that we have an area of high concentration and then an area of low concentration here on the right. And that's a gradient, having an area of two different concentrations. This gradient here, it wants to, what you might say, even out. It wants to become homogeneously mixed, right? So that there is no high and low area, that it's all the same concentration, and all of the molecules, the green molecules, are spread apart in an even way, right? So naturally, molecules move from an area of their high concentration to an area of their low concentration. And that's what this arrow is telling you. This arrow is saying that these solutes, what do you want these to be? Glucose, sucrose, oxygen, carbon dioxide, whatever. These solutes here, the green bubbles, they will just naturally move from the left to the right. No energy needed. This is passive movement. It just happens in the universe. Okay, so now that we understand a concentration gradient, which is 2 points, and those points have different concentrations in them, and we also understand that a concentration gradient doesn't just give you 2 points where there's 2 different concentrations, it also tells you that there will be movement. So that's the important thing about a concentration gradient, is that it helps you determine movement. Now here in this picture, we have obviously a high concentration gradient right here in this area of all the green solutes, but everywhere else in this glass, we have low concentration gradient. I mean, the concentration gradient over here in the lower right-hand corner of this glass is 0. Okay, so diffusion will make this happen. Let's go back and watch that again. Oh, I can't make you watch it again. Diffusion is a movement from a high to low concentration gradient. And you know that if you put ink into a glass of water and come back to it over time, it will become a homogeneous mixture. And you didn't stir it, you didn't heat it, you didn't do anything. The movement of diffusion has no energy required. If we were in the class and someone came in and started to eat an orange, the molecules that have the scent of the orange would start to diffuse throughout the classroom, right? And different people would smell it. Oh, orange, oh, orange, oh, orange. And there's no energy required. The person eating the orange did not blow on the molecules to get them to reach you. So diffusion ultimately wants to make a homogeneous mixture. In fact, when you die, your body, if you're not embalmed, you will decompose and your body will become a homogeneous mixture with the universe. So the fact that you're living means that you maintain these concentration gradients. And when you die, all of your gradients will naturally, with no energy required, make you a homogeneous mixture, but then it will also make you a homogeneous mixture with everything else in the universe. Now, cremation speeds up that process. Just an FYI there. Okay, there's different types of diffusion, right? So there are different ways that molecules can move without energy. Simple diffusion is just the movement between a membrane. This is what we have here. This is a phospholipid bilayer. We have the heads of the phospholipids, they're orange, and you can see the little tails up against each other. And you can see that this picture is showing you that carbon dioxide and oxygen, they both just fit through the phospholipid bilayers. The phospholipid bilayer is kind of like a police line, right? Locking arms. And the carbon dioxide and oxygen just kind of fit right between them. If you're a little person and you've ever been to a concert, this is how you do it. You kind of push your way to the front so you can see. But I know how it is. I'm barely 5 feet tall. All right, so carbon dioxide and oxygen are very small. They're also non-polar. And this is why they can fit right between the phospholipids. They can pass the hydrophilic and the hydrophobic regions of the cell membrane. The picture in the middle here right now, I want you to disregard this just for this slide. That is not simple diffusion. Carbon dioxide and oxygen are showing you simple diffusion. Simple diffusion for us, we want to consider it with a membrane. And that's what you see here. This is a picture of a beaker that has a membrane separating it, and it shows you it from the left to the right throughout time. This is nice. It's a good picture. It gives us a picture of a concentration gradient on the left and a homogeneous mixture on the right. Now, you could consider that the picture on the left is a picture of a cell and its surrounding environment. And it's the same thing. They're separated by a membrane. This just gives you a nice picture here. So how fast will this happen? If I set up a beaker like this with a membrane and a concentration gradient, how long do I have to wait until I can come back and it will be a homogeneous mixture? Depends on how big our beaker is, right? If it's bigger, longer distance for the molecules to travel, it will take more time. Depends how big our molecules are. If we've got something like carbon dioxide and oxygen, not a lot of time. If we have things like glucose, larger molecules like lipids or a protein, that will take more time. If it's hotter, it will happen faster. You know that, right? I like to make hummingbird food where you mix sugar and water, and if I just heat the water up in the microwave first, the sugar will dissolve faster. It will make a homogeneous mixture with the water faster. You also know that you might know, if there's a steeper gradient, it may happen faster. The molecules move faster. Think about this analogy to this as a truck rolling down a hill or a ball rolling down the hill. If the hill is steeper, if the gradient is steeper, then the ball will roll faster. And finally, this is the one that's so difficult for us to realize, but we have to realize this, that in your body, it's not just concentration, it's not just the molecule that makes the difference, it's also the electrical forces. So how many ions, cations, anions, what's in the area? So positives and negatives make a difference. Voltage makes a difference. All right, that was simple diffusion, where carbon dioxide and oxygen just bully their way through the phospholipids. In facilitated diffusion, we are still moving from a gradient of high to low. Check out this picture here. I like this picture. I stole it from the internet somewhere, right? This gradient from high to low, up here on this part of the picture, there is a high gradient. And that's how you can note high. Down here on this part of the picture, there is a low gradient. So that means that all of our molecules, whatever they are, they're going to move from the top to the bottom. Okay, so facilitated diffusion means you're a molecule, you're either too big or you're not charged correctly to cross the hydrophobic region of the phospholipid bilayer. Now, remember, this is in another PowerPoint about cell membranes, but this right here, this is the nonpolar or hydrophobic region of the phospholipid bilayer. And molecules that are polar, like water, can't pass that. So they need a protein. It's kind of like a bridge. They need this protein to span the phospholipid bilayer so that they can use the protein as a bridge to cross. And that's what you're seeing right here with water. Water is crossing over the phospholipid bilayer with a protein called an aquaporin. What type of molecules usually do facilitated diffusion? Polar ones. Water, glucose, ions, right? Like sodium and potassium. All right, so simple diffusion, little tiny molecules, nonpolar. Facilitated diffusion, a little bit bigger, polar. Let's talk about that protein channel for a moment. And I want to tell you that this picture is taken from a larger picture that's in another PowerPoint about proteins and membrane transport. There are different channel proteins, so not all bridges are the same. Some bridges you got to pay. Some bridges you got to wait. There are some channel proteins that are called leak proteins, and as the name indicates, they leak. They always allowed ions through. It's kind of what you see in this purple protein on the left here. There's no gate, there's no nothing, just if you fit, you get through. Kind of like a sink, right? The drain is open, water gets through. There's also protein channels that are called chemically gated. And this picture here has an example of a gated channel. It doesn't tell you how it's gated, but what this means is that you got to pay to cross. You have to have a token to cross the bridge. So if you're a molecule that wants to use the protein, you got to find somebody else with the key to open the protein up. And that's what chemically gated means. It means that there is a molecule somewhere that fits with this protein and then opens the protein up. There's also voltage gated, and I'm just going to mention them, but we talk about them in chapter 12 and on. But there are some proteins that open with a difference in voltage. And this is, again, where we're getting back to those electrical forces. I want to point out that chemically gated are sometimes called ligand gated. If you're reading a website that uses that term, your website is too old. Find another website. Oh, and yeah, the cell membranes mini lecture goes over these protein channels in more detail. Oh, God. And the last thing is, no matter if your channel's opened or closed, it doesn't matter what the gating is on the channel, you still have to have a gradient for movement to happen, right? The gradient determines if and in what direction movement will happen. Not the channel, the gradient. This concept of tonicity can help us understand, or it can help us determine which way molecules will flow, the direction. It will help us determine the direction. Tonicity is the measure of solutes. So you were saying high concentration, low concentration, but we can apply some words to that. A low concentration of solutes is being hypotonic. That's the term, hypo, low, tonic solutes. And you can probably guess that the opposite is hypertonic. Yes. The problem is that these words are relative. They're not absolute. This is like using the terms older and younger, right? These terms are relative. If you refer me to my father, I'm younger than him. But if you refer me to my little nieces, I'm older than them. We'll talk about this in a second. We'll talk about this. So the thing is that in the human body, sometimes we don't use the term tonicity. When we get into higher chapters, we will start to use the word osmolarity. And you're thinking, osmosis? Yeah, we're getting there. Osmolarity is the total solute concentration. So in talking about tonicity, we have to refer to some basic words here that your book uses, I use, and you probably think you know, but you might not specifically know. The word solution for us in this class is going to refer to something that has a solvent that's liquid. And for us in this class, it's pretty much always water. And then a solution refers to something that also has a solute. And a solute can be a solid, like salt, or it can be a gas, like carbon dioxide. Here, water and salt make salt water. That's the solution of life, actually. Here, Princess Leia and water make seltzer. No, I'm sorry. Princess Leia is carbon dioxide. If you push bubbles, carbon dioxide into water, you get seltzer. Seltzer is hypertonic to pure water. If you're to add some salt to that, so now let's add a little bit of salt, a little bit of carbon dioxide, and push it into the water. That's what you get club soda. Club soda is hypertonic to seltzer. Okay, all right. This is the concept that we're trying to get to by understanding diffusion and tonicity. So this, osmosis, is, the pinnacle of our understanding here. Let's not forget some of the basic definitions. Let's define osmosis. Number one, you need a semi-permeable membrane. So talking about the orange that someone might open up in class, the scent diffuses through the classroom, that doesn't have a membrane. That is not osmosis. That is also not osmosis because osmosis only refers to the movement of water. Only water. If you want to talk about the movement of glucose, of salt, no. Only the movement of water. There's a simple way to remember which direction in which water will move. And that's by defining the hypertonic area in any place. Let's just look at these pictures down here very, very quickly. And I want you to see that the little dots are solutes. So the cell on the left is hypertonic. It has a lot of solute in it. And this cell in the middle is hypotonic. It doesn't have a lot of solute in it. So water is drawn to a hypertonic area. It's almost like the solutes draw the water to them, and they do. That's exactly what happens. And what you can hear some people say is they'll say, water follows salt. Yes, there are some drugs that are diuretics, and what they do is they make you release lots of salt into your urine. That's how they work. And then the water follows the salt. And this is a reference to the direction in which water will flow. Water will flow across a membrane to a hypertonic area. Now, I know that some of you are like, wait a minute, Amy, the word hyper, I thought that concentration gradients were high to low, and you're saying that we're moving to a hyper area? Yeah, I am, because the word hypertonic refers to the solutes. That prefix hyper tells me that there's a lot of solute, right? That prefix hypo tells me that there's not a lot of solute. That's not talking about the water. Think about that one. This here, these are real cells. And this is what happens when they shrink or burst. Over here on the left, we have some cells that they've shrunk. What happens is all of the water has been sucked out of them because the environment around them is salty. This is what happens to your cells when you are dehydrated. It's not that you lack water. Dehydration by your body is defined by having lots of salts in you. Have you ever been bloated? You're actually dehydrated when you're bloated because you have so much salt in you. Okay, so the water was sucked out of these. This is called crenation. That's the word that we give to red blood cells when they shrivel. The word over here on the right is just the opposite. These red blood cells are almost about to burst. They have so much solute and salt in them that they're sucking in the water from around them. And when they do burst, that is called hemolysis. Or you want to call it hemolysis? I'm okay with that. Hemo meaning red blood cell and lysis meaning split. That's not really the way you pronounce it, but if we want to do that here, that's fine. What if the concentration inside and outside the cell is the same? That's called equilibrium. And this is when the cells are iso isotonic. Iso meaning same. These little red blood cells here in the middle, that's what they should look like. Nice and round with a little Cheerio kind of center to them. Why do I care about osmosis? Because it's really important for the maintenance of mucous membranes. We talked about these in chapter 1 very briefly. Mucous membranes line cavities that are open to the outside. Take a breath in. You have a mucous membrane along your mouth and your nose. It descends into your throat, down your trachea, and into your bronchi and lungs. Right? That's all a mucous membrane, and it has to stay moist. So in the cells that line these mucous membranes, there's a protein channel. It's called the CFTR. It's discussed in more detail in another PowerPoint. But this channel, it allows chloride ions to flow from the inside to the outside of the cell. Look at this picture on the bottom left. It shows you that. Chloride ions, see the green arrow? Chloride ions are flowing from the inside to the outside of the cell. And guess what follows the ions? Water follows them. And what this does is it makes a watery mucus lining your trachea, lungs, bronchi, and any open cavity, right? This mucus is watery, and it's really easy for it to have movement, and this is the way it's supposed to be. Unfortunately, there is a disease called cystic fibrosis, and it can manifest in many ways, but basically the protein doesn't work. Or, you know what, it might not even be there. Over here on the right, the picture on the bottom right shows you this protein. It's not allowing anything to move through it, right? So this protein as a channel doesn't do its job, no chloride ions, no water, and therefore the mucus that collects on the inside of the trachea, bronchi, and lungs becomes very thick. It doesn't have a lot of flow to it because it's not watery. And these are the conditions of cystic fibrosis, which is a thick layer of non-flowable mucus lining the mucous membranes. Okay, there's just two more types of transport that I want you to look at. Vesicular transport is about making little bubbles inside the cell, and we see a little bubble down there on the bottom right. That's not a cell. It's just a bubble inside the cell. That little bubble or vesicle is capable of transporting lots of ions in one fell swoop. And so this is sometimes called bulk transport. You're not moving a few ions across the cell membrane. You're moving lots of them. This is an exodus of many people. If you are moving solutes out of the cell, which is the opposite of this picture here. That's called exocytosis. That's cell puking, right? Exocytosis, cell puking. If the cell is taking in solutes, that's called endocytosis, or even phagocytosis. That's called cell eating. And then there's pinocytosis, which is cell drinking. Interesting. So if you watch the non-protein organelles PowerPoint, it talks about some things in the cell, lysosomes and peroxisomes, that do phagocytosis and pinocytosis. The last one, is active transport. And all I have to say about this is movement from high to low. This is like pushing a boulder up the hill. This is swimming against the stream, going against the flow, going against the gradient, and this requires energy. So you have to have a protein, can't do it on your own, and you have to have ATP. Look at this picture here. The little green squares are solutes, and they are moving from a place of low concentration to a place of high concentration. That's against the gradient. Do any of you feel like that's like your life sometimes? Most of the time? All the time?
It's active transport. That's what it is. Active transport. Energy needed.